Department of Microbiology with Martin Schuster.
All right, so before I get into what my project was focused on, I'm going to talk a little bit about the bacteria I studied. It's called Pseudomonas aeruginosa, or PA for short. It belongs to the group Proteobacteria. It's rod-shaped, as you can see in the picture. It's gram-negative, meaning that on a gram stain it will appear pink, and it is motile. It's a facultative anaerobe, meaning that although it can survive in environments with oxygen, it can also survive in environments with little to no oxygen. It's an opportunistic pathogen, meaning that when given the opportunity, it will infect a potential host. For example, if you have a severe burn wound or cystic fibrosis or are in other ways immunocompromised, it will infect you given the opportunity. And it's commonly found growing pretty much anywhere from soil to water and even places that are considered more sterile, like hospitals. And a little bit more about Pseudomonas aeruginosa. It produces pyocyanin and pyoverdine metabolites, which combine to form a sort of blue-green pigment when they're plated. And they're resistant to many antibiotics, which make them extremely difficult to kill when they establish an infection. And one of the reasons for this also that they're extremely difficult to kill is that they express valence factors in response to cell density, which is known as quorum sensing, which can have the ability to infect the host quickly and release toxins that will kill the host. They can grow as a biofilm to protect themselves, which is basically sort of layers of Pseudomonas aeruginosa surrounded by slime, meant to protect the bacteria in the center, sort of, and its complete genome has been sequenced. And why is Pseudomonas aeruginosa not some other type of bacteria? Well, Pseudomonas aeruginosa is the main cause of death in cystic fibrosis patients. And once into adulthood, about 80% of all cystic fibrosis patients are infected with Pseudomonas aeruginosa in their lungs. Once again, it's an opportunistic pathogen, and it's a model organism for the sodium quorum sensing a little bit. And a little bit about cystic fibrosis. It's a recessive genetic disease, and it's caused by a mutation in important proteins that regulate the production of mucus and sweat. And most people have at least one working copy of this protein, but people with cystic fibrosis, both of the copies will be mutated. And because cystic fibrosis is incurable, it often results in lung transplantation or death for the patient. And Pseudomonas aeruginosa is related to cystic fibrosis in that it causes fatal infections, which are very difficult to get rid of once they've been established for a long period of time. And now about quorum sensing, this is basically how bacteria communicate in response to cell density to express virulence genes. So you'll see here that signal synthase is producing autoinducers at a fairly low rate in response to the low cell density. But once cell density increases, more autoinducers will be produced by surrounding bacteria, and they'll be able to attach to the bacterium receptor, which causes quorum sensing to activate, which lets the bacteria express their virulence factors. And this can occur between different types of bacteria, not just Pseudomonas aeruginosa. And LASI and LASAR are the autoinducer synthase and receptor of Pseudomonas aeruginosa respectively. And how quorum sensing and Pseudomonas aeruginosa are related is that a quorum sensing proficient population of bacteria is required to infect the lungs of patients. So Pseudomonas aeruginosa mutants without sensing will not be able to infect patients, and LASAR mutants will rise over time in the lungs of cystic fibrosis patients. These are basically Pseudomonas aeruginosa without the ability to quorum sense. And later on in the established infection, the Pseudomonas aeruginosa will convert to a mucoid phenotype, which basically overproduces slime, and this will eventually be fatal for the patient. Very bad prognosis. And my project is basically to discover if there's a relationship between quorum sensing deficiency in Pseudomonas aeruginosa and their conversion to mucoid. And my hypothesis is that there is a relation because of some of the following discoveries. LASAR mutants are more vulnerable to hydrogen peroxide, which is released by the immune system in response to bacteria establishing an infection. And LASR mutant presence in the lungs basically a lot of the time precedes mucoid conversion of Pseudomonas aeruginosa, and hydrogen peroxide stress causes Pseudomonas aeruginosa biofilms to convert to mucoid. And this is important because quorum sensing helps Pseudomonas aeruginosa produce its virulence factors, which help it establish the infection and release toxins to kill the patient. These like attachments, releasing exoenzymes and toxins. And these virulence factors can be turned off by quorum sensing drugs in development, mostly in early cystic fibrosis patients, because once they establish an infection, as I said, they're very difficult to affect. But if you limit quorum sensing in the Pseudomonas aeruginosa, they can begin to behave like LASR mutants, and quorum sensing will turn off, which may, if my hypothesis is correct, make them convert to mucoid, which will kill the patient even faster. And also, it's very important to have a general knowledge of these types of things you're